Okay, so a couple of announcements, reminders. The next exam will be next Friday, not this Friday, but next Friday. So that's the 18th, right? October 18th. So think that day in here, I think there's no one after us, so I can stay a little bit later if necessary. So I think there's someone before us on Fridays. So the earliest we could get in here would be like 10 to noon, which I'll see if I can get in here that early. And then I'll try to stay a little bit later for those of you who could stay right up to 1 or a few minutes after. And then it's going to start with flown translocation and work its way forward all the way to this slide. All right, so the exam material will stop at this point. I think this is, let's see here. On my slide set, it's slide 30. So slides 1 through 30 will be on the second exam, and then 31 forward will be exam 3. So essentially, the bulk of the light reactions and the details of it are not going to be on this second exam. It takes a while to learn that, so we'll put it into the third exam. Questions on the exam content? Or anything regarding the exam? You start your research projects this week. Things are trickling in via mail, so hopefully by this Thursday, you'll have what you need in terms of things that had to be ordered, unless there's some delay due to the storms that have been coursing through the southern states. But that's the plan, to start it on this Thursday. And then I wanted to go over some of the light reactions again to summarize what we've been talking about. Let me go. So this is a nice summary, but it's not all the details. So, but it's a little cleaner and easier to look at for some of the key issues. So we've been looking at this idea how when you think about the light reactions, with plants, you think about the idea that there's two photosystems, and those are transmembrane major big complexes. So you have photosystem 1, photosystem 2, and then cytochrome B6F is also very large and spans the width of the membrane. And then your ATP synthase. So those are the physically big players. And then you have these mobile carriers that will shuttle electrons between these really big players and hopefully keep everything cooking along just right. We also want photosystem 1 and photosystem 2 to be getting hit with the same quality and quantity, well, quantity especially, of light. And that it's the photosystem 2 where you see the oxygen evolving complex as part of photosystem 2. And that is really an amazing process, because the oxygen evolving complex, they named it that because it's the spot where they think water is getting stripped of electrons. And if you do that often enough, you'll end up with oxygen as one of the products. So oxygen evolving. Okay, so the idea here is that light energy is striking both photosystems. And when that happens, if you go into both of the photosystems in their reaction centers, there's a special chlorophyll that its electron's going to get excited and boosted to a higher state. And that's what they're showing here. So once it gets excited, hopefully into the triplet state, it's then called a good reducing agent. That means it's very happy to give off that excited electron. If there's an acceptor nearby, it's good. All right, so it's good at reducing another molecule. And so with this idea, it's going to be pheophytin. We'll get that electron. And then if everything goes okay, what's going to happen then is you have the plastoquinones are next in play. So pheophytin's going to get that electron. It's going to turn, so to speak, and say, hey, plastoquinone. And so there's two plastoquinones. So we label them first and second. The first PQ gets the electron and then quickly gives it to the second PQ. So they call it a 2-electron gate, not because there's two PQs involved, but because that second PQ, in order to do its job, it needs 2 electrons. Plastoquinones just need 2 electrons in order to get their fully reduced form. And so once that second PQ gets 1 electron, it's not enough. It's going to wait until another one comes through. And so you have to go all the way back to the beginning where light energy is striking that chlorophyll in the reaction center, getting the electron excited, given off to pheophytin, given to the first PQ, given to the second PQ. And then that second PQ goes, okay, now I have 2. I'm feeling better about this, but in order to be fully chemically stable, 2 electrons and then 2 protons. And so the plastoquinone will pick up or grab 2 protons from the stroma region. And so now you have protons disappearing, free protons disappearing from the stroma. They're getting attached to plastoquinone. And then we denote this, PQH2 means it's fully reduced. It's got 2 electrons, 2 protons. So we talked about that visually. Here's the idea of 2 electrons, okay, on figure B, where you see 2 electrons and 2 protons are needed to get the fully reduced PQH2. So if that happens, and then that PQH2 is free to do its job, which when you go back to that image, it's moving. It's a mobile carrier. So it's going to release from near, being associated with photosystem 2, and then it's going to move laterally in that hydrophobic portion of the membrane. And it is going to move until it bumps into cytochrome B6F. So now it has 2 electrons that it can donate off into the cytochrome B6F. And so now that is leading to the idea of the Q cycle. So here you have the start of it. So one of the PQH2s releases off photosystem 2, comes along, donates 1 electron into the so-called linear path, and then the other electron goes into the Q cycle. And so those players are going to be a cytochrome B and then another cytochrome B, which then gives it to another plastoquinone. And then that, again, all plastoquinones are like, yeah, I want 2 electrons. So when it gets that 1 electron, that's not good enough. You have to have all of this repeat. And so when we say repeat, we mean you have to go all the way back to photosystem 2, light energy striking photosystem 2. That special electron of the chlorophyll gets excited, boosted to pheophytin, and on, everything repeating, so that you can get another electron coming along with, the mobile carrier PQH2 is going to come along again and donate another electron off to the semi-reduced plastoquinone. And then it's not quite happy yet. It needs 2 protons. So again, we're going to get those from the stroma. So 2 protons are going to be picked up and attached to PQH2. So now they're no longer free protons. So you've essentially made protons disappear, so to speak, free protons disappear from the stroma. So then if you look at the Q cycle in completion, where did it go? Here's the first and second round of it. So here's the first PQ coming along, getting that plastoquinone in the cytochrome B6F semi-reduced. Then everything repeats back to photosystem 2, and you get that second plastoquinone coming along and donating it another electron into the Q cycle. And then you can see where the protons are disappearing, the free protons are disappearing from the stroma. So if you have protons disappearing from the stroma, and then the other thing that you notice going on with the Q cycle, when the PQH2 is donating off electrons, it's also going to dump its protons. And as that's happening, it's in a very specific location. It's in the lumen. And so now you start to see, there's a disappearance of free protons from the stroma. There's, a buildup of protons in the lumen. So you get this gradient of, this proton gradient across the thylakoid membrane. So you can imagine if this is happening over and over and over and over, and the timing of this is phenomenally fast, you can quickly get a nice proton gradient through the Q cycle. So the Q cycle really does help kind of build up the protons. If you were to erase the Q cycle out of the light reaction process, you would see far fewer protons shifting into the lumen and building up. Questions on the Q cycle? Okay. So that's all happening in the cytochrome B6F. But there's another thing that's happening in cytochrome B6F, and that's the start of, or the continuation of, the linear pathway. And so when you look at that one, you have to think about, where did it go? So when that PQH2 is coming along and donates both electrons, one goes into the linear path. And so it gets given to an iron sulfide and then to a cytochrome F. And then, and that's all in the cytochrome B6F complex. And then there's a plastocyanin mobile carrier kind of hugging up to cytochrome B6F, waiting for its electron. And that's going to happen. Cytochrome F will give the electron to plastocyanin. And then when that's done, plastocyanin can disassociate from cytochrome B6F and start moving along the lumen side of the thylakoid membrane. It's attached to the membrane, so to speak, or associated with it. I think I mentioned last time, I always think about those crazy roller coasters where you dangle from the track and zoom along like that, which I would never do, but brave people have done that. And then once you get that plastocyanin approaching to photosystem 1, hopefully everything's going okay with photosystem 1, where it is ready to accept that electron. But you have to have photosystem 1 absorbing light energy and getting that special chlorophyll in the reaction center to get excited. That electron gets excited, and then it gets given off to the acceptor molecule. Then that chlorophyll is missing an electron. Now it's in a state where it can turn to plastocyanin and say, yep, I can take that electron now. And so P700 plus means that that's the chlorophyll, that it's now lost an electron and it's ready to accept one from plastocyanin. So you have to have light energy striking photosystem 1 in order for plastocyanin to do its job. Otherwise, plastocyanin will stall out, so to speak, and just kind of be in neutral until the chlorophyll in photosystem 1 is ready to get an electron. So once that happens, when that chlorophyll in the photosystem 1, its electron gets excited and donated off, these are the players. So the first acceptor within photosystem 1 is another chlorophyll that they've denoted A0. And you can just put A0 if I ask you what's the first acceptor, in the photosystem 1 that's getting that excited electron. So it's another chlorophyll, A0, and then the electron's given to phyloquinone, A1. And then there's an iron sulfur X that passes it to an A, that passes it to a B. And this is being shown here, photosystem 1, they're trying to draw the pathway through the big complex here. And they're trying to show how, plastocyanin comes along and associates and is able to donate off that electron. Okay, so if everything's going okay, it'll pass through all those players in photosystem one until it gets to the ferredoxin. So you can see ferredoxin sitting there, it's associated with photosystem one waiting for that electron. And then if that electron is given to ferredoxin, they're trying to show that it then releases off of photosystem one. And then it's mobile. It's a mobile carrier because its job is to go find the last enzyme in this whole process and give off its electron. And so here, if everything's going to go okay here, you have the ferredoxin, it's going to release from photosystem one. Now it's kind of floating around in the stroma and it needs to find FNR. And FNR is now called the ferredoxin NADP reductase. And so what it's doing is it's taking an electron from ferredoxin. That's why that's in the name. And then the NADP reductase, that's trying to tell you that it has an enzymatic function where it takes NADP plus and gets it reduced. So it's a reductase enzyme. So it's reducing NADP plus. So FNR is like the last player in the game here in order to get NADP plus reduced. And so if we can take that all the way through and get NADP plus reduced, that's great because NADPH is another and very important molecule. That's one of the huge goals of the light reactions. Plants want that because it's called a reducing molecule. It's really good at going along and giving off electrons where needed. And we're going to see how it's needed for the second part of photosynthesis. So what we just described is working along here, the electron flow all the way through photosystem one. So both ferredoxin and FNR, technically mobile carriers, floating around in the stroma, and they'll be interacting with each other. And then the FNR interacts with NADP plus and helps get that reduced to NADPH. And that's a huge goal. That's not the only thing though. The other major goal is to get ATP. And so we've already talked about how the Q cycle is contributing to disappearance of free protons in the stroma, buildup in the lumen. But that's not the only way we get a gradient established. There's actually, that's one big mechanism and it's very important. But the other one is water. Getting water stripped of electrons, one of the products is protons. And so now that's another source of a buildup of protons in the lumen. And so you get all this buildup of protons in the lumen, get a nice gradient. And so they can't go through just the regular membrane. They have to go through the ATP synthase. And that provides the torque force energy to get the ADP phosphorylated. So then that's the other huge goal. So now they've made the NADPH and they've made the ATP. And both of those products are showing up in the stroma. Very important that you know the locations because it really kind of makes sense because the next phase of photosynthesis, it's all happening in the stroma here. And so to have the products right there where they get fed into part two of photosynthesis is a good strategy. Okay. Long process, many steps that need to go and repeat, go all the way back to the beginning. So it's a complicated process. This is the more detailed version of all the players in the game, and they put it onto this energy tower, so to speak. And so for the details of it, this is a really good slide to think about. One other concept I wanted to mention. So when you peer into the chloroplasts and look at what they're doing, most of what they're doing are the light reactions, as we just went through them. However, some sections of the thylakoid membrane of some of the chloroplasts they might kick into this idea, cyclic photophosphorylation. And again, it's not all the time. And it's happens in some plants more than others. And it's an interesting process. So it's cyclic. So what they mean is the electron flow, instead of going linear from water all the way to NADP plus, that doesn't happen. It's a circular flow. And so I've drawn this trying to show the cytochrome B6F is this like oval shape here. And here's your plastoquinone coming along from photosystem 2. And then what they're trying to show here is that the electron flow goes to photosystem 1 to ferredoxin, but now nowhere does this show that ferredoxin is giving off the electron to FNR. It's not, the FNR is not a player here. So it's not, ferredoxin gets the electron, and instead of turning to FNR and saying, here, take this and give it to NADP plus, it turns around and goes back into a circular flow where it'll come back, the electron flow comes back into cytochrome B6F. And then back to PQ. And then you can get this, and it's usually just in one section of the thylakoid on some of the chloroplasts where the electrons will zoom around like this in a circular path. And here's another way to think about it in kind of context of we're trying to show plastoquinone here. Here's the plastoquinone coming along and bumping into cytochrome B6F. And they're trying to show that, you do get to photosystem 1 to ferredoxin, but then it comes back. So if you look at the way they drew this, do you see any NADPH showing up? It's not there. So the artist is correct by leaving NADPH out of the picture here. That's accurate. But you do see the other major product of the light reactions. You still see ATP showing up. So what the artist is trying to show here is that if portions of the thylakoid are allowing for this circular flow like this, it still gets you ATP. No NADPH, but ATP. Because it's still allowing for this proton gradient. When you have plastoquinones donating off, going into this Q cycle of the cytochrome B6F, you got all that going on. And so it's kind of like a ramping up of the Q cycle and getting the proton pumping across the thylakoid membrane. So you get a nice buildup. So in essence, what it's doing is it's augmenting proton pumping and it's allowing for more ATP production. So if a plant is under some kind of issue, some kind of thing going on, it's in its environment where it's forcing it to work harder, so to speak, where like, oh, geez, I need more ATP. Then it can do this. It goes, okay, I need more ATP. I'm going to ramp up into cyclic photophosphorylation and some parts of my thylakoid membranes. And that will kind of give me this boost of extra ATP. So like I said, it doesn't happen all the time. It's more of a as needed basis, but it is a very quick and easy way to just kind of beef up the proton gradient and get a steeper gradient, so to speak, and allow for some extra ATP. So keep that in mind. I'll come back to this idea because I'll explain what kinds of plants might be doing this and why, and put it into more context when we get to that section, which is coming up soon. So basically, it's taking the plastoquinone to the cytochrome B6F, plastocyanin, photosystem 1, FD, ferredoxin, and then it comes back. And it just keeps going round and round and round and round. And as you see, when you do that, you get proton pumping across the membrane, proton pumping across the membrane. And so it's a pretty slick way to ramp up the ATP. And so when you do this, you get extra protons getting pumped. It's a very efficient, everything's already there in place, just kind of ramp it up a little bit. And you don't get NADPH, but that's okay. You don't get that idea, but that's okay, because sometimes it's just like, no, I don't need any more NADPH per se. I just need a boost of ATP. Okay. And so that is something that will do. So this all has to, if we think about photosynthesis and all of the players, all the things that have to go right, light energy has to strike both photosystem in 1 and 2. And then you think about all the things that can mess with it. Like you already messed with it in lab when you, for example, expose those chloroplasts to DCMU. That's an herbicide, and you were messing with the light reactions. Specifically, you were messing with the plastoquinones. Ability to transfer electrons. You can see, so DCMU they actually know exactly what part of the light reactions you're impacting. And then paraquat is another herbicide. And you can see that one is toward the end of the light reactions where it messes it up. Some plants are more sensitive to these herbicides than others. So there's lots of things that can go wrong, including damage. Sometimes you'll get some aspects of the photosystems damaged, for example. And basically, if you've ever heard your parents say too much of anything can be bad for you, it's true with plants as well, and light. If they get too much light coming in too fast, where their light reactions can't keep up with it, it can actually create bad situations, including free radical formation. And so they refer to it, if it's too much light energy coming in too fast, they refer to that damage as photooxidative. Basically, what that's saying is that you get, instead of an excited chlorophyll donating its electron off into the light reactions, it starts to go, it messes up and will give electrons off to other things instead. And then you can get essentially free radicals. Often it's a singlet oxygen that's highly reactive. In general, they're toxic chemicals, free radicals. And then once those get produced, they start causing significant damage to the proteins of photosystem 1 or 2, for example. Lipids, the thylakoid membrane is made up of a bunch of lipids, of course. So it can be kind of this domino impact, like dominoes, one thing falling after the next, where things can get really, really damaged. So it makes sense that if a plant has to deal with light, that's a lot of energy coming at you, if they have to work with light on a daily basis, they need some way to protect themselves and from too much. And so there's several options. One of them are those carotenoids. So as far as we know, every photosynthetic organism has carotenoids. And they think the reason for that is because they offer photoprotection. They help protect the plant from too much sunlight. And so they often refer to the carotenoids as safety valves for the plant. And they often use this term: it quenches the excited state of chlorophyll. So if those chlorophyll molecules get too excited, where the electron transport essentially starts to fall apart and get out of whack, the carotenoids, essentially what they do is they will take on some of that excitation and essentially there's a little reaction that they can run, and it allows for the extra energy to essentially, through that process, get converted to heat energy instead of excited electrons that go on to be given to oxygen and creating all these free radicals. Safer to deal with heat. So if you can take that excitation energy and create a situation where heat energy is the result rather than free radicals, you'll pick the heat energy. So these carotenoids seem to do that. Here is a flow chart where they've kind of mapped out some of the processes they know plants can do. So here they're showing, too much light energy coming in, and then they show that heat idea here. First line of defense, try to convert that excitation energy to heat energy. And then you can see that you can get the green box are all the free radicals that can form. And then they're saying a couple of things here. Well, let me go forward once. So let me finish the thought on carotenoids. So xanthophylls are a type of carotenoids. And so what they see is that when you have too much light energy, this is trying to show that it's a short little reaction. The violazanthin to anthraxanthin to zeaxanthin. You don't need to memorize that. What I'm trying to show you is that you get this little reaction, and as these are all carotenoid pigments, as these pigments are doing their reaction, it releases heat in the process. So this is why they say it's like a safety valve for excess light energy. If you wanted to visualize it through the reaction pathway, that's what it looks like. Again, I'm not going to ask details on this, but if you like a visual idea here of how, yes, they're definitely going through a reaction pathway. And as that's occurring, it allows for heat energy to be given off. So carotenoids are super important. It seems like every single living organism that does photosynthesis has carotenoids. The other idea here, they show in this flow chart, so to speak, is the damage to photosystem 2, and they pinpoint a very particular protein called D1. So that's a protein that's part of photosystem 2, and they're showing that it gets oxidized, and then it leads to photoinhibition. That just means that you have enough damage to photosystem 2 that photosystem 2 doesn't do a very good job anymore of doing photosynthesis. And so for some reason, the D1 subunit of photosystem 2 seems to be like the weak link. That's the first thing that will get damaged when they run experiments in the lab. They see the D1 kind of being the first to fall. And so they refer to that as photoinhibition, because essentially you damage photosystem 2 enough that it can't really perform correctly for doing the light reactions. And when they do this in the lab and they play around with it, they can see that photosystem 2 can recover initially with a little bit of damage. It's like, yeah, I can, recover that. They can actually show that the D1 will get damaged first, but if it's a low enough damage, the plant will actually take out the damaged D1 protein, make a new one, and stick the new one in place. So it can cut and paste and try to repair that. But if you keep in lab experiments where if they keep bombarding that photosystem 2 with just keep doing too much light energy, and they keep doing that, then it's permanent. And you will have photosystem 2 essentially start to fall apart. So this is just a picture of photosystem 2 showing you there's lots of different subunits, different proteins involved. And so they've done enough research where it's the D1 that seems to be the weakest link. And then they've played around with photosystem 1, of course, trying to figure out what could go wrong. And some of the studies are showing that free radicals, active oxygen species, are free radicals. So when ferredoxin gets its electron in the light reactions, it's supposed to give it to FNR, but sometimes ferredoxin has a whoops and it doesn't, and it will actually give the electron to oxygen, which is nearby, and that will yield superoxide, which is a free radical, and which of course is going to start damaging membranes and proteins. And so the plant has certain enzymes that can take and work with the superoxide and reduce it to less damaging forms. So superoxide dismutase, you might have heard that, or ascorbate peroxidase. So these are enzymes that the plant has. A lot of aerobic organisms like us and plants have these enzymes because we have to deal with reactive oxygen all the time. You're dealing with it right now, and is something that is going to be impacting you and your tissues and cells throughout your whole life. And so that's why they say you want to have antioxidants in your diet, because you're trying to nip this in the bud. Just, similar to what the plant is trying to do. Okay, so if everything's going okay and they have like carotenoids up and running and they have these enzymes available, hopefully all of the damage can be minimized or non-existent. The other huge thing though that the plant deals with is that, with photosystem 1 and photosystem 2, and remember there's lots of them embedded in the thylakoid membrane of a single chloroplast, you gotta have the same amount of energy getting into those photosystems in order for everything to go in concert or in step. So you have a really nice electron flow. And that can be hard to do if you just think about the complexity, especially like I always think about big trees where they have big canopies. You have lower branches, middle branches, upper branches. The upper leaves are getting hit with full-on sunlight. Some others are getting shaded out. You have all of the nuances of the chloroplast might not be getting optimal and equivalent light energy coming in. So hard to do. So the plants have evolved mechanisms for this too, where they can shift their energy. It's pretty amazing. And so if they detect one of the photosystems needs a boost, they can allow that boost to happen. So what happens, so LHC, that's the antenna system around photosystem 2. So LHC2 is around photosystem 2. What happens is if a protein gets phosphorylated, so we're talking ATPs going on here and donating off a phosphate group. If that happens, the LHC2 will actually move to photosystem 1 and deliver its energy to photosystem 1 instead of photosystem 2. So that phosphate that gets stuck onto the antenna system is the signal. Hey, photosystem 1 needs some extra energy. And so it will actually shift and give energy to photosystem 1. It actually physically moves, which is kind of bizarre. And so you have this the LHC2 antenna complex. If it's in its phosphorylated state, if that happens, that's the signal. I got to go find photosystem 1 and give extra energy there. So the way that this decision gets made, should we phosphorylate LHC2 or not? The way the plant is assessing this and thinking through this, so to speak, is that it's dependent on, so just to clarify, if there's no phosphorylation, it just stays around photosystem 2. But it's dependent on PQH2 levels. So the plant is assessing how much PQH2 do I have? Do I have a lot? Not very much. And when it's doing that, that is the signal to the plant whether or not to phosphorylate this antenna complex. So basically, if the plant detects, oh boy, I have a lot of PQH2. So that's the reduced form, PQH2. So what that's saying is there's lots of reduced PQH2 building up. How would that happen? If you have a lot of PQH2 building up, so what that's saying is things are cooking along quite nicely on this end of the photosynthesis. So photosystem 2 is doing its job, getting the electron to feel phytin, to plastoquinone, and you get PQH2. So that's going along and it keeps going. But if it's building up, what that's saying is down the line, something's going wrong because it stalls out. So somewhere forward of PQH2, there's a problem. And that's usually right here. Light energy is not keeping up with photosystem 1 compared to photosystem 2 when the light energy. So you have a difference in light energy striking these two photosystems. If you have a lot of light energy hitting photosystem 2 and not much with photosystem 1, that means that you won't have photosystem 1 doing much of anything, which means PQH2, it's like it's getting backed up. The PQH2 can't donate off its electrons because everything is stalling out further down the line. That makes sense. So if you have that situation, what the plant is queuing in on is how much PQH2 do I have? And that, if it's saying, wow, I have a lot of PQH2, it must not be able to donate off its electrons because further down the line it's stalling out. That's the signal. Okay, I'm going to phosphorylate LHC2, which tells it go find photosystem 1 and get it kicked into high gear. And then things will even out and you'll get the flow all the way to completion. So they're paying attention to some of the details of what's going on with the light reaction and using that as a cue to send a signal to the LHC2. So pretty slick little mechanism. It works. The photosynthesis, when it's cooking along, is really efficient, amazingly efficient for a biological process this complicated and this massive of a job of stripping electrons off of water ultimately and getting them all the way down to NADP plus. Okay. Oh, this slide is just comparing. A lot of students might be more familiar with like aerobic respiration going on inside mitochondria. So I just put this in here for those of you who've taken like animal or human phys and have worked through that process, or maybe biochemistry, where you look at what's going on with the electron transport chain in the mitochondria. And you can see that there's definitely some similarities here in the overall process and goals, which is to get plenty of ATP in that process. Okay. All right, we've worked through all of the light reactions from start to finish. This will not be on the second exam because it's a lot. All right, this is a lot. When we think about learning this process, I expect the details. All right, so as you're working through this slide is a good one. It gives all the major players and will show the electron flow. This one is nice for kind of summarizing and giving you the idea of who are the physically big versus the mobile carriers, that kind of idea, the stroma versus lumen, proton gradient, all of that. Okay, so I think these two slides are really, really valuable as you're picking it apart and learning the steps. Okay, but I do expect detail for this process. Okay, so as you're studying this, if you are unsure, like, how much detail, just ask me, either email or see, in person.
So these two slides won't be on the exam, the next exam?
No, it takes too long to learn this. I want to give you time. It's a process. So we're going to stop at, what was it, slide 30 or something like that. This goes, 97 slides. So we're going to do 1 through 30 for the next exam. Other questions on the light reactions? Okay. Well, we'll stop there for today. The next thing we're going to get into is, okay, so they've done the light reactions. They get all this ATP, they get all this NADPH. Now what? So we're going to talk about what happens to those. And basically that's getting into how does the plant make their sugars? So we'll do that on Friday.